And we are honored to have Dr. Marcy MacDonald here for our closing keynote. After Marcy, we'll also have Jeff and Etta. We're honored to have Marcy. Marcy has a whole list of credentials here. Marcy obtained her PhD in medical biophysics at the University of Toronto. Marcy's Canadian. I hope Marcy doesn't mind me saying this, but I wanted Marcy's mother here so she knew that Marcy has a real job. But she couldn't make it to join us. Maybe next time, because we'll get Marcy back. Marcy is currently a professor of neurology genetics at the Harvard Medical School and the Massachusetts General Hospital. You may recall yesterday that Dr. Jeff Carroll did his postdoc working with Marcy. And I said, boy, we're getting lots of brain power here. One thing that's really pretty incredible, and having Marcy come here and talk to us today, is Marcy was on the team that found the HD gene. Marcy's one of the researchers that you have to go up to her and say, "We love you." Marcy?
And my mother would be happy to know I have a real job. There's no security, but that's just, one of those things. I want to tell you all that you are trailblazers and pioneers, as if you didn't already know that. You are all part of a worldwide HD research enterprise. It sounds sort of like the Starship Enterprise. It's really meant that way because it is a global network, and everybody who is an HD family has a chance to participate in this research enterprise. And many, many of you already do, and hopefully many more of you will also into the future. And I'm going to explain what I mean by that in this talk, because you're a pioneer. HD is out in front. You may not realize it, or you may. There are many, of course, brain disorders, but there hasn't been a true disease-modifying therapeutic found for a brain disorder. And so you guys are out in front. We together are out in front in showing the other disorders how to do this. And we can do it because Huntington's disease has the same genetic root cause. And believe it or not, that gives the research enterprise a leg up. And so that's my job today, to show you how we're pioneering and trailblazing. So what's research? It seems like a no-brainer, but just to put us onto the same page, this is what I mean by research. And the nature of research has inherent to it the idea that for any given step, when you have an idea and you're trying to test it out and see whether or not the idea is going to stand up or get knocked down when you know something in addition to what you knew before, it means it takes time. And the reason that that is important is because you really want to build a solid foundation. You don't need to know everything. So, for example, there was a time when people in the world thought that the Earth was flat. Did not have to wait until satellites and spaceships and things outside the atmosphere to actually show everybody a picture of a round Earth. In fact, the ancients figured it out long before by looking at the heavens. So observation and research could tell people it's got to be round, even though there was no telescope from space looking back at the Earth to say that it is round. And that's what we need to do with HD research. We don't need to know everything. We just need to know enough that what we are doing serves as a solid foundation for things going forward. Why do we want that? Because we don't want to be guessing. We don't want to have to play hunches. And in HD, because there's a single genetic cause, we don't have to play hunches. All the causes of Parkinson's disease are not known. All the causes of Alzheimer's disease are not known, or ALS, many, many, many other disorders. The cause in HD in every case is known. We can start there. Why do we want to build this foundation? Here are a bunch of reasons, and I think they all speak to the research enterprise. People think of research as being something that people do with test tubes and animals and stuff in a laboratory. It's only one little segment of it. There's lots and lots of information generating that is research that goes into driving these things forward. You need solid information because that's what patients and families deserve. You should be making decisions based on information that next week is going to give you the same idea as you had a month ago. You need it to be solid and enduring. Similarly for disease management, you don't want to be doing things that are going to make things worse simply because somebody thought it might be a good idea. You really would like to have solid information on which to make those kinds of decisions. And developing therapeutics, obviously, and we're going to talk a lot more about that. But also, design of clinical trials. Clinical trials are designed now using measurements that are really pretty rough. And so what we need are better ways to measure things in patients that will empower the design of clinical trials, the number of people, the kinds of people to be in the clinical trials. You can't make those decisions if you don't know what the disease process is and how it looks in patients. So this is, in essence, a rough schematic of how you would go about, or rather, how the current way of going about getting a therapeutic using research, as opposed to guessing. You start with a molecular target, and therein lies the rub. You have to have the right target. So lots and lots of discovery research goes into figuring out what's the right target. Is this target still going to be right at 2 years from now when we know a lot more? Or is it going to turn out not to have been a right thing? So lots of research goes into that. And the whole sort of left-hand middle side of this schematic has to do with discovery, what people would call basic science research. The middle overlapping with that, going off to the right, is applied or clinical research. And you need it all. And I'm going to try and explain a little bit about what it all is. The basic discovery that that you heard Bev mentioned was the research that cloned the Huntington's disease gene, because before that research, it wasn't known. Why was that worth knowing? Well, because that would provide a place to start on this whole arrow going from left to right. And before that, there was no place to start that made any sense. It was just throwing darts at biology and saying, oh, I think this might make a difference. Information building. That discovery wasn't done in a vacuum. That discovery needed a whole bunch of other things. I've just listed a few here on the left-hand side. One was a description of Huntington's disease in families, saying that it was inherited, that in essence it was genetic. In 1872, even before it was recognized that there were things called genes, or indeed a thing called genetics. What he said was, it didn't skip generations. Well, that's inheritance. And he said, if it isn't in a generation, then it isn't going to come back. That's inheritance. What's DNA? That wasn't really understood until 1949, the chemical nature of it and its structure until later in the 50s. But the important finding that motivated people to do that biochemistry and all that basic laboratory research was the finding, after lots of arguing for a very long time, about what the material of inheritance actually was. And there were a whole series of experiments to actually prove that it was DNA was the entity that described inheritance. It didn't matter if you put proteins into things, it was the DNA that empowered the cell and gave the inherited trait. So the HD gene discovery and the finding of linkage using genetic analysis in patient families built on that prior knowledge. And the linkage set up the knowledge to be able to find the HD gene and the mutation itself. That in itself, as I just said a few minutes ago, now opens up all kinds of doors and things that you need to know. Research as well as better disease management, including a direct test for the CAG repeat that is the cause of every case of Huntington's disease. And you're all familiar with this, that the gene turned out to be the HD gene, now called HTT, because the nomenclature committee changed the name after the fact, as committees are wont to do. But it's still the same gene, it's still the same DNA, and the mutation is still the same. It's a CAG triplet repeat that encodes a glutamine residue in the protein called Huntington. And that knowledge of those DNA sequences allowed the development of a very specific test for clinical application to determine how many CAGs, and much research involving a lot of HD families and patients came up with the finding that everybody has HD genes, two in fact, one from your father and one from your mother, and we all inherit two HD genes in all of the cells of our body, and each gene has a different number of CAGs at that position, and that's called a polymorphism. And so in the general population, the number of CAGs in that tract of CAGs can vary from about 6 to about 34. From 35 to about 39 is what's known to be reduced penetrance. Some of you will be familiar with that because in this area, some individuals may go on to develop the overt symptoms of HD while others will not. And then finally, there's the fully penetrant range in a genetics term, which means repeats over that number inevitably go on to give overt clinical symptoms of Huntington's disease in a normal lifespan. So this allows clinical testing in various scenarios, some of which have been discussed here at this meeting, confirming a clinical diagnosis, pre-symptomatic diagnosis, prenatal, pre-implantation diagnosis, and others, as giving people information and choices that are suited to their lives. The cloning also showed that the HD gene is expressed in all cells of the body right from conception. That sets up the expectation that the gene can be doing its damage right from the get-go, and that it's very, very subtle. It takes a very, very, very long time for this very slight difference in DNA sequence to wreak havoc. So that means what researchers have to look for in their discovery experiments has to be something that is subtle and CAG length dependent. This is the goal. Now you know on the left-hand side at the bottom, you know what the trigger is in all cases. Now the goal is to figure out what is the disease process, what are the molecular, cellular events that emanate from that trigger in all kinds of cells in the body, especially in the brain, that get the cells and the tissue, especially the brain, to the state where it no longer can compensate, and functionally it just has to give up the ghost. And so now there's overt clinical symptoms because, for example, the brain has just completely run out of capacity to deal. That's clinical onset, clinical diagnosis. But that typically is decades and decades and decades. Not always, but in many, many cases. That means that there's a really big window to be able to modify potentially a number of those steps along the way, those molecular things that determine what's happening in the future, if they can be identified, as well as identifying things for once the functional capacity is no longer there, how do you give the brain neuroprotection? How do you have what's left work better for longer? So after onset, also symptomatic treatment, as well as disease-modifying treatments. There are lots and lots of basic and applied research studies that are going on right now. I've only listed a very few of the observational studies that we'll talk about in a minute. But since 1993, this graph has been populated with hugely important studies that are designed to leverage information in the basic discovery at the bench mode, basic research, as well as the clinical applied research, which comes from observations in people. So let me describe some of those. Those are the two kinds of research right here. So one of the questions is, what does the HD gene expansion do in cells that have the expansion that's different from cells that don't have an expansion? That's a basic discovery research question. A clinical applied question, also related to observational studies, would be, what's the earliest thing I can measure in somebody who has an HD expansion that would distinguish them from somebody who has a CAG repeat that's shorter? So that's also a research question. And it's also an important question because if you could know with certainty that you could measure in that thing in somebody before they had overt clinical symptoms and had to go to a neurologist, that would allow you an opportunity to try something to intervene before the stage of having to go to a neurologist. So those are both research questions. They're both important questions. Basic research mostly is discovery. It's mostly around this single question. What's the target? What's the best molecular target from over there on the left-hand side of the therapeutics arrow? How do we choose the best targets? How do we find them? How do we test them and make sure that they're solid? Well, a lot of this research happens in model organisms where you can put the gene into the organism or generate it so it has exactly the same mutation, ask what the consequences are with different repeat sizes, look at the molecular level and the cellular level and how do the cells function, what cell type is important, what cell type is the most sensitive, and so on. And then you can build up a picture biochemically. What does the Huntington protein do? What does it look like? What's important for it to really give cells that toxic push? And then you need to synthesize this information in a way where you actually come out with understanding that you didn't have before. Because it's no good having a bunch of different narratives that don't really connect to the same end. So a lot of effort goes into having the research studies actually coordinate and be interpretable together with a common language so that understanding can flow from all of this research. Why does it take so long? The goal is to go from the mutant gene at the bottom of the slide, building a foundation that's completely solid, starting from that point, all the way up to the top of the slide to that yellow brick right at the top, which is a therapeutic target that, when interfered with, will work, is the prediction. And so you have to put all those bricks in place, and you have to see that the bricks actually hang together, and that there's a therapeutic target that looks like it's on a solid foundation below. This is how research actually works. You put a few bricks together, and you think, I'm doing fine. Everybody else who reads your papers gives you money to fund your studies. Other things thinks, oh, you're doing fine. Until you do that experiment that says, oops, nope, that wasn't predicted, shouldn't have seen that. That shouldn't have happened, can't be right. And that's how research works. And that's why it takes quite a long time, because the time is an investment in that solid foundation rather than the single narratives that may not go anywhere because they're not truly connected to the HD gene and the goal of the therapeutic target. That said, there are an awful lot of things that look really good right now, and that some of them are pretty far along. And they fall into sort of three basic categories. The first category is the biggest. This comes from the discovery research in animals, animal models as well as biochemistry, and cell models. And there are two approaches basically that are around actually trying to do something about the HD CAG repeat expansion and the HD gene itself. And the whole idea behind this is because this expanded copy does something extra in addition to what the other copy does, it would be great if you could know what to measure so you could tell that you were doing something. But if you could knock it down, that expanded copy, ideally just the expanded copy, or make the CAG repeats smaller in the expanded copy, in the cells in the brain, for example, you would imagine that that push on that disease process would now be slowed down because you wouldn't have as strong a CAG push. That is, I think, one of the most exciting things that is the farthest along. The top one, the zinc finger endonuclease, which would make the CAG repeat shorter in the cells, isn't so far along, but there's traction there. The one that's farthest along is the next one, and it's an example, is synthetic oligonucleotides. There are other approaches to knock down the expression of the expanded gene. These are quite far along. And these are looking very, very promising. And there are biotech and pharmaceutical companies that have partnered with foundations like your own, like CHDI, the HDSA, the European agencies, and CHDI has really brought a lot of focus into this area. So that's a big one. The other way to go about it is to say, all right, I know what the mutant gene is doing to cells, and I know it in enough detail that if I identify something that interferes with that thing, it will make the cell better, or at least it won't make the cell get worse. That's the modulate the early consequences of the gene approach. And there are many areas I've listed for energetics, transcription, that is, regulate the cell in a different way, synaptic function, how do the brain cells talk to each other, clearance of toxic material. If you can help the cell out because it's struggling, that may be having a therapeutic benefit. And there are biotech and pharmaceutical companies who have already invested in this research, and that's a really good thing. The other general area I mentioned before is neuroprotection, which is, okay, maybe you're not going to stop the disease process, but what can help the brain cells function better? And if we knew what those things were, maybe there's a way to apply those. And so that's the neuroprotection area. And there are lots of groups working on this as well as biotech and pharmaceutical companies. The final areas are really nascent, and those are leveraged by stem cell research. The one I'm most excited about is using induced pluripotent stem cells from adult skin cells, for instance. And I'm sure you've probably read a lot about this. So these aren't embryonic cells, but they're from skin cells from adults that can be reprogrammed in a dish. And this can now be done with HD patient skin cells, so that you can now turn them into neurons and brain cells in a dish and study them with the actual HD mutation in them that has caused HD in the patient. That's hugely exciting, and that's discovery research. But there's a therapeutic element to this that isn't well developed and is far off, in my opinion, down the road, that relates to stem cells, and that would be stem cell replacement, for instance, and also stimulating new brain cell development from the stem cells that are already in adult brains. So those are far down the roads, but those are ideas that people are working on. The applied research is really important because, as I mentioned earlier, you have to know what to measure in somebody who has a CAG expansion, and you need to know when to measure it. Otherwise, even if you had the perfect therapeutic target and the perfect therapeutic, how would you know it was working? You need to know what to measure, which means studies looking at what the repeat does in people who have the repeat. And I'm delighted to say that the research community has finally come to the conclusion that the patient is king. Mice are nice, but the patient is king. So there is a lot of HD research now going on with HD patients, their cells and their tissues, as well as these iPS cells that I mentioned earlier. Some of you may have participated in some studies. The first question that was answered with studies like this is, when can you first pick up changes in the brain if you just look with brain imaging? And the answer is you can pick up changes pretty early, even before clinical onset, diagnosable symptoms. Regions of the brain here in green, these are just scans, they're pretty. The regions in the green have changed a lot. These are taken in patients who haven't yet had a clinical diagnosis. And in fact, you can do lots of measures. They're just graphs of measures. You'll notice on all of them, either going downwards from left to right or going upwards in the top left-hand panel. About 15 or so years before a clinical diagnosis, you can actually make measurements that says people have lost the capacity to do this very specific task. It's not enough to diagnose HD on clinically, but it could be enough to actually go in with much better measurements in an observational trial or a clinical trial and take a look and see whether having given somebody something 10 years or 2 years or 3 months earlier might have improved that measure. And that's 10 or 15 years before onset of clinically diagnosed symptoms. I mentioned the stem cells. Lots and lots of studies on induced pluripotent stem cells, which opens up a whole new window, including the window of you can turn these cells not only into brain cells, you can also turn them into other cells of the body. Maybe there are things that can be measured in liver cells, skin cells. You don't have to measure it in the brain. We don't know. And hopefully these studies will help people understand that. There are genetic studies, another curve, which says that for any given CAG repeat length plotted along the bottom, onset of clinically diagnosable symptoms can occur around a mean or average that's plus or minus 18 years. Lots of modification. People have inherited other gene variants that are influencing when they get clinical symptoms with a given CAG repeat. So that's another way to find modifiers that may affect different steps along the disease pathway because people with the same CAG have onset at widely disparate ages. Those are modifiers. If we knew what they were, and from genetic studies we're trying to find that, we would have something, potentially, that is capable of modifying the key disease steps in people. This is a worldwide effort. Many of you will have heard of and or participated in PHAROS or PREDICT, cohort, track. registry. These are observational studies that collect specimens and information. And these have been so successful, they have actually motivated a much bigger study going global called Enroll HD. And you will have potentially seen some of the materials here at the desk outside. These are incredibly important. The previous studies were and still are incredibly important because information generated in all of those, in individuals at risk, as well as individuals with clinical symptoms, that information doesn't go away. That information can be used from here on out. Every time we add another brick, we can go back to that information and say, does this new brick fit with what we knew before? Enroll is aimed at gathering even more information, similar kinds of information, but basic information worldwide. There are sort of, if you take a look at Enroll, a bunch of different things that Enroll can do. There are too many to name. These are some of the ones that I think are the most important things to come out of Enroll for research. That is clinical information and biospecimens, blood, tissue, various other things from each individual worldwide, as many people as possible, symptomatic, not symptomatic. It serves as information that can invigorate brand new studies on patients, and who knows, maybe even on mice. But studies in mice that are related now to studies, to things that are actually observed in patient cells and tissues and in patients themselves. It also acts as a way to really speed enrollment in clinical trials. And this is very, very important, obviously. You don't know where you are in building that pyramid until you can figure out if you are still building toward that pinnacle, or are you off on a little garden wall somewhere over there on the right. And there are clinical trials. Enroll and all the other studies aim to build better clinical trials, better targets through information and research. But these are ongoing, and these also generate information. The important thing about a clinical trial is that it is done in a way that the information is meaningful. A clinical trial with no follow-up to find out how the person who took the intervention is doing isn't useful. It's a one-off. That information isn't going to help anybody. So these kinds of clinical trials are important, because if it turns out that the dose isn't the right dose potentially, or the agent that's being tested isn't necessarily doing what the people thought it might do, that information is completely needed, because that's how research works. You have to know if you're off building a garden wall or whether you're still on a main path. So, lots and lots of things have happened from 1872 until today, and each thing builds on what went before. And in this information age, let me tell you, that's not always easy to keep track of. And in fact, I think you could look at all the researchers in the room and they would have to admit to you that they couldn't possibly read everything that's written on HD now. It's really huge and good. So it's hard to remember what we didn't know before, because now you know it. But remember that, because when you get impatient, that's part of the issue. All the researchers want to thank HD families, caregivers, clinicians, other researchers. You will recognize many faces, and there are many, many folks in Canada, around the world, who do HD research, some of which the HSC helps to fund and get off the ground, for sure. Including, do you know who the gal on the bike on the bottom right is? Does anybody recognize that researcher? Any guesses? If I said Eileen, would that spur anybody's imagination? That's Eileen Donovan Wright. And I think that's one of the few times that she's been outside of the research laboratory. But she was caught on camera riding a bike. But there are many, many researchers. And this effort is not possible without HD families, clinicians, caregivers, time, effort, and money. Now, we still need your help in all of those ways, but we also need your help in a very important way. I believe that we scientists do a great job at many, many things, and we do some things very badly. One of the things we do very badly is communicating what we find and why we're trying to find it and what it means to somebody. So we need your help in building an infrastructure and, in fact, an expectation for clear information coming from the research, coming from the scientists, whether it's a clinical scientist or a basic bench scientist, all scientists, because we are innately, most of us, incapable of doing it without a great deal of prodding. And so one way that you can do this is to demand it. And these guys have created a place for you to complain. I don't know if they presented this, but let me tell you, write that URL down that's in the blue at the bottom of the page. And you hit that and you email them and go to the site and you say, I didn't get it. What the heck? Email them. Email them your ideas about what you don't get. I just read in the newspaper this story that this was the greatest thing since sliced bread last year, but now it's not. How the heck could that happen? Why don't you guys do a story on that? Enlighten me. I can't be the only person that has this question. This is your chance, and this actually is groundbreaking. There is no place for clear scientific communication between scientists and the patients and families that they purport to want to help. And I think we do. It isn't just purporting to. We actually do. We're just not good at explaining it. Here's your chance. Get it from the horse's mouth. There's a horse. It takes a community. It takes a community of wonderful clinicians like Dr. Ed Wild, who I would never kid, ever. And it takes patients, and it takes families, and it takes researchers, and it takes clinicians. You really are part of a global HD research enterprise. And we are very glad that you are. Thank you.